Dear organizers and colleagues, thank you for the opportunity to speak at this conference. And let me give you a short introduction into my most recent research on bacterial oxylipins. Oxylipins are oxidized derivatives of polyunsaturated fatty acids, which are synthesized by various enzymes, including lipoxygenases, or LOX. In a previously published preprint, we presented a data analysis of lipoxygenase protein sequences deposited in publicly available databases. We performed blast search, phylogenetic research, selected the lipoxygenases of pathogens and symbionts, non-free-living bacteria, and used their species names as PubMed search queries. Then the resulting abstracts were used for network text analysis, where nodes are terms characterizing the ecophysiology of the respective bacteria, and the edges were co-occurrences of the terms in the same species. We have found that for lipoxygenase positive bacteria, it is very characteristic to affect plants as symbiosis or as pathogenesis, affect humans, especially patients of hospital, nosocomial pathogenesis, and cystic fibrosis patients. And in some species, these functions are overlapped, as you can see by a lot of connections between green part and the red part. Some bacterial species of this dataset can simultaneously affect plants and humans, including cystic fibrosis patients, and some of them are emerging pathogens with antimicrobial resistance. Here you can see the graphic ecological portrait of lipoxygenase positive bacteria created on the basis of the network presented on the previous slide. In our literature review, we noted a similar role of lipoxygenase and C-FTR inhibitory factor, or CIF, of Pseudomonas aeruginosa in the colonization of the respiratory tract of cystic fibrosis patients. This similarity seemed very interesting to us, and we decided to perform a similar analysis for CIF homologs. But we faced a much lower quantity of available data. Despite this, we performed a phylogenetic study of the homologs of Pseudomonas aeruginosa CIF, and using another experimentally characterized CIF of Netobacter nosocomialis, shown here as a road mark, as a mark to a calibrator analysis, if I can say this way. We assumed that all this cluster consists of functional CIFs of bacteria. We extracted species names from this cluster and performed a similar literature review, systematic review of PubMed abstracts. But this time without network analysis. But in this table, you can see similar association. A lot of human opportunistic pathogens, a lot of plant pathogens, a lot of bacteria with antimicrobial resistance, and some bacteria, a lot of bacteria, have overlapped functions between affecting plants and affecting humans. So the ecological characteristics of these enzymes, CIF and lipoxygenases, are similar. And it can seem strange, but CIF had previously been characterized as an epoxide hydrolase that cleaves polyunsaturated fatty acids epoxides and synthesized the immunosuppressive oxylipins. The reference is included here. So we can assume that oxylipin spoofing is a universal strategy of bacteria to make cross-kingdom host jumps and affect patients with cystic fibrosis. And the similarity of ecological characteristics of these bacteria might indicate a functional relatedness between CIFs and lipoxygenases. Thank you for your attention. Please ask your questions.